Hello, everyone, and welcome to today's live broadcast, the STREPTAG System for Efficient Protein Purification of Expi Supernatants, presented by Dennis Karthoff, Scientist, IBA Life Sciences. I'm Alexis Krauss of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, please visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. Please join me in welcoming Dennis Karthoff. I will now turn the presentation over to him.
So thank you very much, and a very warm welcome from my side to all of the audience. Today I would like to introduce you to the STREPTAG system and how you can use it for efficient protein purification from Expi supernatants. So in life sciences, we have many applications and experiments where we need only protein in a milligram range, but we need it very quickly. One example is the drug discovery process of pharmaceutical companies, where often several hundreds of possible candidates are expressed and screened simultaneously. And in this phase of R&D, the pharmaceutical companies try to reduce the time as much as possible, because the earlier you find a leading candidate, the earlier you can go on with your clinical trials, and finally, the earlier you are on the market with a product that brings in some money. Another application example where milligrams of protein are sufficient are toxicological studies or binding and kinetic studies. And furthermore, for first animal studies, usually a few milligrams of protein are sufficient. In basic research, you're often dealing with many different projects, but again, there you often need only protein in a milligram range, but you need it very quickly because you don't want to waste much time in the production of your protein. You want to start with your experiment as quick as possible. Another experiment is crystallography, where you usually are fine with a few milligrams of protein, or the assay development, and finally protein interaction studies. So when you rely on a mammalian expression system, transient expression is the best way how you can produce protein in a milligram up to gram range for some proteins in a very short time. Because you don't have to screen or to select stable cell lines, you can reduce the time for the production, and usually you get your protein within days. So in transient expression, you benefit from a very high initial expression during the first days after transfection before the expression drops. And that makes transient expression a perfect choice for high throughput applications like drug screening. And we already heard some nice presentations today where such applications were outlined. So the Expi Expression System from Thermo is a mammalian expression system that is very well suited for this transient expressions. There are two systems available on the market, the Expi 293 system and the Expi CHO system. And both systems come with chemical defined media that allow the cells to grow up to very high cell densities and perfectly match transfection reagents, enhancer solutions, and feeding solutions. So due to this high cell densities and the achieved high protein titers, it's possible to reduce the scale for your production and therefore the footprint in your incubators. And that allows you to simultaneously screen more drug candidates or process more proteins in parallel. And that's why this system is very often used in pharmaceutical companies for their R&D and drug discovery applications. However, expression of your protein is only one side of the coin, and the other one is purification. And every process is limited by its bottlenecks. So in order to have a benefit of such high protein expressions and the ability to screen many different proteins in parallel, you would need a purification strategy that is capable of purifying so many different proteins simultaneously. The best way to do this is the use of tag systems, because tags can be fused to any kind of protein, and this allows you to purify many different proteins with the same purification strategy and the same materials, which will save you a lot of time during the downstream process. Not surprising, there exist many different tag systems on the market, and here are just a few examples of the most prominent ones. One group of tags are so-called epitope tags, like the flag tag or the MIG tag. These tags rely on an antigen-antibody binding, and therefore they are often eluted at low pH values, which can affect the properties of your protein. In addition, the resins usually have a comparable low binding capacity and are quite expensive. And this makes purification, especially of larger amounts of protein, expensive if you use these tag systems. Another group of tags are large tags like the maltose binding protein or the GST tag or the streptavidin tag. This tag can increase the solubility of your protein of interest or its expression, but on the other side, these tags can also influence the properties of your protein of interest, leading to a different behavior in your further experiments. And that's something, of course, you would like to avoid. In addition, due to their high size, they are highly antigenic, and if you want to go with your protein into some first animal studies, you usually have to cleave off these tags before you can go on with your experiments. So the most widespread tag system on the market is the HIS tag system, because the system is cheap and the tag is small, so in many applications it's not necessary to cleave the tag. However, even this very widespread system has many disadvantages. First of all, and we've already heard it in the talk of John, the purification of mammalian or sometimes even insect cells can be quite challenging, because mammalian cells have more host cell proteins with histidine residues that can be co-purified, and chemical-defined media that allow the growth of high cell densities often have interfering substances that interfere with the HIS tag purification. In addition, customers are often facing inconsistent purities, which make it necessary to do a second purification step in order to get protein purity that is sufficient for your experiments. Furthermore, the tag system often needs a kind of optimization, for example, the optimization of the imidazole concentration in the sample or the wash buffer, in order to get a good compromise between purity of your protein and protein yield. And finally, the binding affinity of the system is not high enough to really efficiently use it in immobilization experiments like SPR studies, because you can see a remarkable dissociation of the tag itself, and that complicates the data analysis. So in this conference, we heard a lot of the Expi system, so this experiment might be very interesting, because here we see what happens if you load Expi supernatant without any further treatment on a common nickel anti-A column. In this experiment, we spiked 20 milliliters of Expi supernatant with an M-sherry fused to an HIS tag and purified this 20 milliliters over a 1 milliliter nickel anti-A column. And as you can see, during the loading procedure, the protein starts to leak from the column, indicated by the white area in the upper part of the column. So it seems that there are some substances in the media that interfere with the HIS tag binding, and obviously this is not the most efficient way to purify your protein from the supernatant. So with all these drawbacks of the aforementioned tag, this raises the question if there is an attractive alternative technology that can be used. And for this, we developed the Strep-Tactin STREPTAG system many years ago. And let's have a brief look how the system works. The system depends on two components. The first component is the STREPTAG peptide, which is an 8 amino acid long peptide that can be fused to your protein of interest. The second component is Strep-Tactin, which is a re-engineered streptavidin that has an increased binding affinity to the STREPTAG compared to streptavidin. And what happens, you can use this tag system in order to purify your proteins. You use your supernatants, for example, from mammalian cell supernatants or bacterial lysates, whatever, and apply it to a chromatography purification column, like an FPLC column or a gravity flow column. And your protein of interest will be bound via the Strep-Tag2 peptide, which is bound to the biotin binding pocket of the Strep-Tactin. Then you can wash away the impurities under physiological conditions, and then you can elute your protein by just adding this thiobiotin to your wash buffer. This is very beneficial because this is a very mild elution, and you don't have to change buffer conditions to low pH values or to high salt concentrations. And that means that proteins that maybe are unspecifically bound to your matrix will not be co-eluted with your protein because you don't change the buffer conditions. After elution, you can regenerate the column by adding vitamin H, which turns the column's color red, and then you wash away the HABA and you can go for a new purification round. So the advantages of the system are high protein purities of up to more than 95% due to these two specific purification steps, the specific binding and the specific elution. Furthermore, you have a great flexibility regarding your buffer composition. The system is compatible with high salt concentrations, detergents, metal ions, chelators, and reducing agents, which means you can adjust the buffer to your protein of interest. But of course, it's also very effective to purify proteins under physiological conditions. The resin is reusable and the tag is very small, so it's usually not necessary to cleave off the tag. However, this system has also one drawback, and this is a relatively low binding affinity in a micromolar range. So the system works very well for bacterial lysates, but when it comes to low concentrated protein samples, which we are often facing when we do mammalian expressions, then this binding affinity can be too low for an efficient purification. So that's the reason why we further developed the system by the development of Strep-Tactin XT and the Twin-Strep-tag. The Twin-Strep-tag is a tag that consists of two Strep-Tag2 peptides that are linked together. And due to this linker, it's possible to target two biotin binding pockets of the Strep-Tactin instead of only one, creating an avidity effect that increases the binding affinity of the system. The second improvement was the development of Strep-Tactin XT, and you can see the structure on the right side, by mutation of Strep-Tactin. And Strep-Tactin XT has a much higher binding affinity for the STREPTAG and therefore for the Twin-Strep-tag compared to the Strep-Tactin. So together, the STREPTAG and the Strep-Tactin XT will have a binding affinity in the nanomolar range, so it's comparable to the HisTag system. But with the Twin-Strep-tag system and the Strep-Tactin XT, you will get a binding affinity in a picomolecular range. And this is the highest binding affinity available among all affinity purification systems that are available on the market. So when we look to the purification cycle of the Strep-Tactin XT, this is quite comparable to the Strep-Tactin purification cycle. Again, you immobilize your protein of interest via the STREPTAG or the Twin-Strep-tag on Strep-Tactin XT. And after you load your sample on a chromatography column, you wash away the impurities, and then again you have this mild elution by adding biotin instead of this thiobiotin, because the binding affinity for biotin and that thiobiotin of Strep-Tactin XT is reduced compared to Strep-Tactin, and therefore we need to use biotin, which will block Strep-Tactin columns irreversibly. Strep-Tactin XT columns can be regenerated after the biotin bounds to the biotin binding pocket of Strep-Tactin XT by just adding NaOH, and then you can use the resin for a new purification round. So now let's have a look to some benefits of the system. In this experiment, we compared the purification of Strep-Tactin high capacity and Strep-Tactin XT high capacity. The high capacity materials have a threefold higher binding capacity compared to the non-high capacity materials. And for this experiment, we used M-Sherry lysates from E. coli with a Twin-Strep-tag and purified the lysates on the two columns. And in the STS-page deal, you can see the load in lane 1, the wash fraction in lane 2, and the elution fraction in lane 3. And if you see the elution fraction, you can see that both systems generated high purity of nearly 100%. The lower band is a fragment of M-Sherry, so it's not an unspecific contamination. However, if you look to the wash fraction of Strep-Tactin, you can see that protein of interest was already washed from the column during the wash step due to the lower affinity. Instead, when you use Strep-Tactin XT, you can see that there are no bands in the wash fraction, in the last wash fraction. And that shows you that protein that is bound to the Strep-Tactin XT column will not be released prior to elution. This is a great benefit over other tag systems like the HISTAG system because it allows you to wash with large wash volumes to even get rid of high concentrated contaminants without losing protein during the wash procedure. The binding affinity of Strep-Tactin XT also increases its dynamic binding capacity compared to Strep-Tactin. And if you look to the right side, you can see the comparison of dynamic binding capacities for M-Sherry measured on Strep-Tactin and Strep-Tactin XT high capacity. And you can see by the use of Strep-Tactin XT, we can increase the binding capacity close to 2-fold times compared to Strep-Tactin. Given to the fact that the Strep-Tactin XT materials are cheaper than the Strep-Tactin materials, it's possible to reduce the cost for a purification by more than 50% if you use Strep-Tactin XT instead of Strep-Tactin. So, I've already mentioned that the system is quite compatible with different detergents, and these data here were provided by Q Biotech, and they purified GFP Twin-Strep-tag fused proteins in the presence of 2% of different detergents. And as you can see in the STS page here, none of the tested detergents had any negative impact on the protein purity, which was nearly 100%, compared to the non-detergent containing control. Furthermore, none of the tested detergents had any negative impact on protein yield, as you can see on the right side, and I think that demonstrates that the system is quite compatible with many detergents and makes it a very good system for the purification of membrane proteins. In this experiment, we compared the purification of HISTAG protein on a nickel anti-A column and Strep-Tactin XT purifications using the same proteins fused to a Twin-Strep-tag. So we used M-Sherry and GAPDH lysates and applied them on a nickel anti-A column and then purified them according to the manufacturer's protocol. And as you can see in the elution fraction of M-Sherry, there are lots of contaminating bands still present in the elution fraction. And obviously here a second purification step would be necessary in order to achieve a good protein purity. The GAPDH purification looks better, especially because the GAPDH is highly concentrated, but again, especially in the upper part, you can see many contaminating proteins. In contrast, if you use Strep-Tactin XT, you can see that we achieve nearly 100% pure protein for the M-Sherry purification. And on the right side, you can see the Western blot that detects both bands, proving what I said, that the lower band is just a fragment of M-Sherry also tagged, and therefore this special case we cannot remove this fragment. And even if you look to the GAPDH, which was not so bad in the HISTAG purification, you can see that the purity is much higher in the Strep-Tactin XT system compared to the HISTAG system. And all these experiments here were done under physiological conditions. So I've mentioned the high binding affinity of the Strep-Tactin XT system, and these data are provided by the company Dynamic Biosensors. And Dynamic Biosensors developed a switch sense technology that can be used for kinetic statics, like SPR, but it can be used for many different other applications. So it's a very powerful technology, and they already released a Twin-Strep-tag or STREPTAG capture kit that allows you to capture STREPTAG proteins on a Strep-Tactin XT chip. And they compared the binding affinity of GFP fused to a Twin-Strep-tag and GFP fused to a STREPTAG in their system. So if you look to the left graph, you can see that both systems have a very fast association rate, but when you look to the dissociation on the right side, you can see a remarkable dissociation for the STREPTAG protein, which has a comparable binding affinity like the his-tag. So if you would use a his-tag capturing kit, you would actually see such a dissociation. In contrast, if you look to the Twin-Strep-tag immobilized protein, you can see a very stable signal over a long time, and this demonstrates that this technology is perfectly suited for protein interaction studies, even if your binding affinity between the two investigated proteins, like antigens and antibodies, is very high. And if you compare the binding affinities, you can see that the STREPTAG and the his-tag are in a low nanomolar range, and that the Twin-Strep-tag Strep-Tactin XT system is in a low picomolar range. So, let's get back to the initial start of the presentation. When we see the problems you can have purifying his-tag proteins from Expi supernatant, and the question if there is an attractive alternative. And when I was on a conference, I talked to Henry Chu, the product manager for the Expi system, and we discussed the topic about the problems with his-tag purification, and he was wondering if our STREPTAG system might be would face the same problems, or if it would be beneficial for the purification of proteins from Expi supernatants. So we started a cooperation, and I'd like to thank Temo for this cooperation in providing supernatants for us, and we started with Expi 293 supernatants. Let me say some words to the experiment itself. All experiments were conducted, if not mentioned otherwise, 2 microliter Strep-Tactin XT gravity flow columns. So the resin volume was very small, and as we are not able to adjust the flow speed in gravity flow mode, the contact times were very short, around about only 20 seconds. Usually for FPLC applications, we recommend 2 minutes. However, we have seen that the Expi system is often used for high throughput applications where only very small expression volumes are used, and therefore you would need small columns, and we thought that this is a realistic application and scenario for Expi supernatants. All proteins that we spiked to the supernatants were Twin-Strep-tag proteins. Okay, so the first question that was very interesting for us was if the biotin in the Expi 293 medium would interfere with our protein purification. Nearly all chemical defined media for mammalian cells contain biotin, and you remember biotin is a substance that we use for elution of our protein. So usually it's not a very good case if you have the substance that you use for elution in your sample. However, our elution buffers have much higher biotin concentrations than the biotin concentration is in the Expi 293 media. And so we wanted to answer the question, do the concentrations of biotin in the media interfere with the protein purification? For this experiment, we used the Expi 293 supernatants. The supernatants were generated by transfecting Expi 293 cells with an empty vector. We also included all the enhancing solutions, and when the viability dropped to 75%, we harvested the supernatant that we used for spiking. So here we spiked M-Sherry and the monoclonal antibody to the supernatant, and then we splitted the supernatants. One part of the supernatant was directly loaded on our Strep-Tactin XT gravity flow columns, and the other part was treated with BioLog solution and buffer W. BioLog solution is a solution that blocks free biotin in media, so the biotin cannot bind anymore to Strep-Tactin XT. However, it would be an additional preparation step that we would like to avoid. So we purified the supernatants and finally analyzed the yields after elution. And if you see the left graphic, you can see that the addition of BioLog and buffer W does not change the binding capacity, and that proves that the presence of biotin in the media does not interfere with our Twin-Strep-tag Strep-Tactin XT binding. And that means that you can directly load your supernatants on the column, which is a great advantage compared to the histag purification we've seen at the beginning of the slide, of the presentation. We also checked the influence of the protein's purity on the right side, and you can see that the addition of BioLog and buffer W does not have any influence on the purity. We were able to achieve nearly 100% pure protein in this single purification step in all experiments. However, another question that was interesting for us was, are there any substances, the enhancer solutions or host cell proteins that maybe interfere with the binding, reducing binding capacity, but not completely inhibit the protein binding? And to investigate this fact, we used pure Expi 293 medium, we used supernatant and supernatant where we added enhancer solutions and spiked M-Sherry protein to the supernatant, purified all samples and measured the eluted amount of protein. And as you can see in the graphic, none of the tested components had a negative impact on the protein yield. That proves that neither the Expi 293 host cell proteins nor enhancer solutions or components of the Expi 293 system do interfere with our Strep-Tactin XT purification. So once we figured out that the system is not a problem for the purification of STREPTAG proteins on Strep-Tactin XT, we wanted to further investigate the performance of our Strep-Tactin XT with the Expi 293 system. So one question was, how is the maximum protein binding capacity on column is influenced by the concentration of our protein of interest in the supernatant? For this question, we spiked 3 proteins, M-Sherry, a monoclonal antibody, and secreted alkaline phosphatase to Expi 293 supernatants to a final concentration of 100 micrograms per milliliter or 200 micrograms per milliliter, and purified the samples and again measured the eluted amount of protein. You can see that for the M-Sherry and the secreted alkaline phosphatase, the protein concentration had no impact on the protein yield. But if you look to the monoclonal antibody, you can see that the binding capacity was reduced as the protein concentration was lowered. This is not very surprisingly because we adjusted the supernatants to the same mass concentrations of our protein of interest. But as the monoclonal antibody will be 2-fold larger than the secreted alkaline phosphatase and 5-fold larger than the M-Sherry, that means that the molecular concentration that drives the diffusion force is lower compared to the other 2 proteins. Furthermore, the monoclonal antibody has a larger size, so the diffusion into the pores, especially to the inner parts of the core, is slower and maybe limited for the protein, for this protein size. And that means that the yield for low concentrated samples are reduced. In addition, please keep in mind that we have here only a contact time of 20 seconds, which is really short for an affinity purification process. And if you have a longer residence time, then you would also achieve higher binding capacities. However, if you look to the protein yield on the right side, you can see that the protein concentration, the supernatant of our protein of interest, do not have an influence on the protein purity. Again, for all 3 proteins, we were able to achieve nearly 100%, at least more than 90% pure protein in a single purification step. Without any adjustments of the purification protocol, it's the same protocol you use for bacterial lysates, etc., so you don't have to optimize the procedure. So if you're purifying protein, usually you don't want to overload your column and lose protein, and therefore we did this experiment where we loaded the columns with up to 75% of the maximum binding capacity we've determined before. And again, we spiked the supernatants with our 3 proteins to the 2 concentrations of 100 and 200 micrograms per milliliters, and compared the amount of loaded protein to the amount of protein that we eluted from the columns. On the left side, you can see that the concentration of proteins in the supernatant had no big influence on the recovery for all proteins, and we were able to achieve very good recovery rates, close to 100% for the M-Sherry protein and 80 to 90% for the monoclonal antibody and secreted alkaline phosphatase. And not very surprising, the purity of the proteins were very high again for all concentrations, as you can see at this page here on the right side. So I think these results showed you that the STREPTACTIN XT Twin-Strep-tag system is very efficient in the purification of proteins from Expi 293 supernatants. Finally, we wanted to make an experiment, you could say, under real conditions, because we have the Expi 293 system here in-house. So we transfected Expi 293 cells with a vector coding for secreted alkaline phosphatase, or the monoclonal antibody, and cultivated the cells until they reach a viability of 75%. And then we harvested the supernatant, which was only 30 milliliters, and purified the supernatant on a 1 milliliter STREPTACTIN XT high-capacity gravity flow column. We used the high-capacity column here because we knew that secreted alkaline phosphatase is expressed very well, and we expected a huge amount of protein, even from the 30 milliliters, and we wanted to be sure that we get all the protein. So you can see when we calculated the yield for 1 liter production, we achieved 300 milligrams per liter for the secreted alkaline phosphatase and 86 milligrams per liter for the antibody. It's worth to mention that this was a rabbit antibody, and we've already heard in the presentations of John that human antibodies are higher expressed in the Expi 293 system, so this is a kind of challenging monoclonal antibody. And if you see on the right side, you can see again that under these real conditions, we were able to achieve very high protein purity from the purification of the samples. So as you can imagine, we were quite happy with this result, and Thermo was too. But then Thermo told us, okay, maybe now we should have a try on the Expi system, because this system is much more challenging. The cell densities are higher, so we have more whole cell protein in the supernatant, and we have a different biotin concentration. So we wanted to see how we can work with our system in this expression system. The experiments were conducted in the same way like for the Expi 293 system. We used Twin-Strep-tag proteins. Again, we used the small column volumes of 200 microliters and therefore the short residence time. And the supernatants were provided by Thermo. It's worth to mention that the supernatants already contained an overexpressed monoclonal antibody that was expressed in the labs of Thermo. So in this supernatants, we have a kind of artificial high concentrated contamination that we usually would not have in an ex vitro supernatant when we purify Twin-Strep-tag proteins. So first of all, again, we checked if it's necessary to add biolog solution or buffer W to the ex vitro supernatants, or if we can directly load our supernatants that we spike with some sherry and monoclonal antibody on the column. And again, quite similar to the results of the Expi 293 system, you can see that the addition of biolog and buffer W did not change the protein yield. So again, also for the XPCHO system, the presence of biotin did not interfere with the binding of our twin-strep-tag protein on Strep-Tactin XT. And if you look on the right side, you can see again the SDS page deal of the purifications. And if you look to the elutions, you can see that even for higher host cell protein concentrations, we achieve very high purities of close to 100% in the XPCHO system. However, if you have a closer look, you can see in the elution fraction 2 residual bands that correspond to the molecular weight of the overexpressed antibody that is very nicely seen in the load of the samples. So, as I've already mentioned, usually you would not have such a high contamination in your supernatant, and obviously our standard protocol with 5 wash volumes is not large enough in order to get rid of such a high artificial contamination. In order to prove this assumption, we used here a supernatant where we spiked secreted alkaline phosphatase to the XPCHO supernatant, and in the SDS page gel in the mid, you can see the Comassi gel of this purification. You can see that we have the 2 antibody bands in the elution fraction of the secreted alkaline phosphatase elution, and we plotted the supernatant and this elution fraction on a Western blot and detected the blot with an anti-human antibody conjugated to HRP. And you can see that the heavy chain of the antibody was detected in the load and in the elution fraction, which proved that this is a contamination you usually would not see in your twin-strep-tag purified samples. However, we thought this would be a great opportunity to see how we can use our system to even deal with such high concentrated contamination. And you remember that we have the possibility to do this intensive wash procedures due to our high binding affinity, which is not so easy with other tag systems. So here we shoot a little bit with cannon to birds, but we purified the 3 proteins, the secreted alkaline phosphatase, the M-sherry, and the monoclonal antibody by applying 30 column volumes of wash buffer during the wash procedure. And if you look to the elution fractions, now you can see that we were able to remove the contaminating bands of the overexpressed antibody. And we think that this experiment really shows that you can even get rid of very high concentrated contaminations, and this is a great advantage of this system compared to other systems because it gives you this possibility to do this intensive wash procedures. Furthermore, you can imagine that you might have proteins that be on the column that you have to wash away by adding some detergents, other by salt concentration. So it gives you also the possibility to wash with different wash buffers without getting in danger to lose your protein, to achieve high purities, even for very challenging purification tasks. So back to the experiments that we've already conducted for the XP293 system. Here for the XPCHO system, again, we wanted to see how the protein concentration affects the binding capacity. Again, we spiked the XPCHO supernatants with our 3 proteins to final concentrations of 100 and 200 micrograms per milliliter, purified them on the 200 microliter XT columns, and measured the amount of eluted protein, and the results are comparable to the XP293 results. You can see that for the M-sherry and the secreted alkaline phosphatase, the concentration of the protein has no influence on the recovery, but the monoclonal antibody binding capacity was again reduced if the concentration was lowered, obviously due to the same effects that I've already explained in the XP293 system. However, at this point of time, we were wondering how the binding capacity would increase if we increase the residence time to 2 minutes, like you would use on an FBLC column, or that you see if you use a column with with a larger batch volume. And for this, we used the Gilson SPAC pipetting devices that allowed us to adjust the flow speed and the residence time. And you can see how the binding capacity changes if you increase the residence time. All proteins we achieved higher binding capacities for all proteins, like 5 milligrams for the M-Sherry and the secreted alkaline phosphatase, and the monoclonal antibody was spiked to 100 micrograms per milliliter in this experiment because obviously this was a more challenging condition, and you can see that it's also possible to increase the binding capacity for this low concentrated monoclonal antibody. And again, on the right side, you can see that the protein purity was not affected by the concentration. The only real contamination you can see is the overexpressed antibody. Because of this, in the following experiment, we haven't used the 30 column volumes of wash volume to save a little bit of time. So again, the next experiment was to check how the recovery is influenced by the protein concentrations in the Expi system. And again, we see that the recovery was not affected by the protein concentration in the supernatant. Doesn't matter if we use 200 or 100 micrograms per milliliter of our 3 proteins in the Expi supernatant. We achieved a recovery of around about 90% for M-Sherry, but you can see that the recovery was lower here, a little bit below 80% for the monoclonal antibody and the secreted alkaline phosphatase. And again, we were wondering how this would change if we increase the residence time. So we did increase the residence time again. And you can see that by increasing the residence time, we increase the recovery for the monoclonal antibody to up to 100% and the secreted alkaline phosphatase to close to 90%. The M-Sherry recovery was not affected, but this is obviously due to the fact that the recovery rate was already very high and was 90%. And again, you can see in the elution fraction of the SDH shield that for all concentrations and all purifications, we get this nice high protein purity. So we have the Expi system here in us, so we're not able to express our proteins in the system itself, but we use the chance to challenge the system a little bit more by testing very low concentrated protein concentrations in the Expi supernatant. And in this experiment, we spiked the Expi supernatant to a final concentrations between 13 and 20 micrograms per milliliter with our 3 proteins. And then we loaded 50 milliliters of Expi supernatant on only 300 microliter bed volume containing Strep-Tactin XT column. And this is a large volume of supernatant compared to the reference volume. Remember the purification of the histec protein, where protein started to leak from the column after already 20 milliliters on a 1 milliliter columns. So here we wanted to see what happens if we apply large volumes, if you use the Expi system for manufacturing of large-scale expressions with our system. Again, we measured the loaded amount of protein and the loaded amount of the eluted amount of protein, and you can see that even for this very small concentrations, we were able to achieve around 100% recovery. It's worth to mention that in this experiment, we used again the ASPEC pipetting device in order to achieve a residence time of 2 minutes. And if you see to the right side, you can see again that the only present contamination is the overexpressed antibody. We use only 7 column volumes for washing the column. And I think this demonstrates that even for very low concentrated sample in challenging Expi supernatants, it's possible to achieve high protein purity and recovery with the Twin-Strep-tag Strep-Tactin XT system. And due to the very easy protocol, I think this system is really perfect addition to the Expi system, and together it's a great technology and system in order to do high throughput purifications of proteins. So, and that brings me up to the summary. The Strep-Tactin XT Twin-Strep-tag system, I think, is really a superior purification system because we have this exceptional high binding affinity in the picomolecular range, and we have an outstanding protein recovery and purity. We can achieve up to more than 98% pure protein with short tag 8 amino acids in case of the STREPTAG2 and 28 amino acids if you use a Twin-Strep-tag. And we know that for many applications this tag does not to be cleaved off. Even animal studies were conducted with Twin-Strep-tag proteins, and that was not a problem. You don't have to optimize the protocol like for the HISTAG system where you have to adjust the imidazole concentration. It's the same protocol for the XP293, for the XP system, for bacterial lysates. So it's a very easy, straightforward protocol. We have a very efficient purification from both XP293 and the XP supernatants. We achieve this high efficiency without any manipulation like dialysis steps or adding of any reagents to the XP supernatants. We can directly load the supernatants on our columns, and we have a convenient immobilization of twin-strep-tag proteins on surfaces like SPR chips or the switch-sense technology. This is very great because you can use the tech not only for purification. It's the first time now that you can use the purification tech for SPR applications, even for very high binding affinity kinetic studies. Otherwise, you have to biotinylate your protein and use, for example, streptavidin chips, but then you have an additional biotinylation step, and I think this is really a great advantage of the system. And finally, we have a high compatibility with many reagents like high salt concentrations, up to 5 molar of sodium chloride, for example, of detergents, metal ions, chelators, and reducing agents. And so, to come to the end of my presentation, sometimes a picture is worth 1,000 words. You remember our nickel anti-A column from the beginning of the presentation that you can see now on the right side with the protein that leaks from the columns. And of course, that was only half of the truth, because right next to this column, we placed the STREPTACTIN XT high-capacity column where we loaded 20 milliliters of Twin-Strep-tag M-sherry to the column. The concentration and protein amount was the same than in the nickel anti-A purification. And you can see that here no protein leaks from the column. We get a really nice sharp binding of the protein in the upper part of this column. And I think this really demonstrates how beneficial the Strep-Tactin XT Twin-Strep-tag system is compared to the HISTAG system. And with this, I thank you for hearing and listening to this talk, and open to some questions.
Thank you, Dennis, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, why does biotin in the medium not interfere with the binding of the STREPTAG if biotin is also used for elution?
That's a very good question. So what happens is the STREPTAG and also the Twin-Strep-tag will dissociate from time to time from the biotin binding pocket. But in case of the Twin-Strep-tag, the second tag will be back in place before the protein is eluted from the column. So if you use biotin in the elution buffer, what happens is in the moment where the STREPTAG will dissociate from the streptactin, the biotin will get into the biotin binding pocket, blocking it for the STREPTAG, and the STREPTAG will be eluted. However, this binding is not irreversible, which means that also the biotin will be released from the biotin binding pocket of streptactin. So you need a large excess of biotin in the elution in order to be sure that the next biotin molecule will go into the binding pocket before the STREPTAG can go back in place. However, obviously, the biotin concentration in the Expi media are much lower concentrated, and there is not enough biotin present to get into the free binding pocket before the STREPTAG gets back into the binding pocket. And you've seen the quick association rate of the STREPTAG in the slides for the switch-sense technology, and that's the reason why the biotin concentration in the Expi and XP293 media are not high enough to prevent the Twin-Strep-tag from binding to the Strep-Tactin XT.
Is it possible to purify very large proteins with the Strep-Tactin XT?
Yes, that's definitely possible. We actually have customers who purified a 1.2 megadalton protein complex with our resin. Of course, you will lose binding affinity because such large complexes will not enter the pores of the resin. But yes, it's definitely possible to purify even very, very large proteins and protein complexes with the system.
Okay, Dennis, our next question is, what are the con and cough rates of the Twin-Strep-tag to Strep-Tactin XT?
Yes, the K-on rates are quite comparable between the Twin-Strep-tag and the STREPTAG to STREPTACTIN XT. So the K-on rate will be in a range of 5.5 multiplied with 10 high 5, and the K-off rate will be 3.3 multiplied with 10 high minus 5 seconds. So the K-off rate is really, really slow, and that leads to a final KD in the low picomolecular range.
Which products can be used for the purification of proteins? Do you offer FPLC cartridges?
Yes, there are several products available, so we have a great flexibility how you would like to purify your protein. You can use FPLC cartridges. We offer 1 milliliter and 5 milliliter pre-packed cartridges, but you can also buy free slurry to pack your own cartridge with the amount of slurry you need. We are also having magnetic beads with STREPTACTIN XT for very small batch purification, and we can use this very efficiently for high throughput screenings of small proteins, of small samples. Yes.
Our next question, is the STREPTAG equally efficient on the N and C termini of the protein?
Yes, usually it is. Actually, this always depends a little bit on the protein itself and how the structure of the protein is, because if one of the termini is folded in a way that the tag accessibility is very low, then of course you cannot bind the tag very efficiently, but then it's a good choice to test the other terminus where the tag accessibility might be better. But if there is no protein conformation that drives the tag to a site where its accessibility is not very good, then it doesn't matter if you fuse the tag to the N or C terminus, it works both.
Did you do tests to optimize the residence time and optimal binding and supernatant volume dilution effect?
I'm not quite sure if I understand the question correctly. So we optimize not the residence time. In most of our applications, the 2 minutes residence time is fine. If we have larger proteins like the monoclonal antibody, larger times might be beneficial, but for most proteins, 2 minutes are sufficient in order to get a good purification. Regarding to the volume of the supernatant and the volume of the resin, it depends only on the binding capacity. So if you have a very low concentrated protein, you can load 2 liters of your supernatant on a 1 milliliter column, but if you have a very high expression, of course you would overload your column.
Our next question is, did you carry out same experiments with magnetic strep beads?
We haven't tested the magnetic strep beads with the Expi system yet. All these experiments were done with the agarose beads, but that's a very interesting question, and I think that gives us a very great idea what can come next. And I think we definitely should look in this in the next experiments, because you can use magnetic beads very well in high throughput processes, but I'm quite confident that this will work too, because the STREPTACTIN XT immobilizer is of course the same than on the agarose beads. But yes, that's something we will have to look in the next experiments.
Can I use this product in an E. coli system?
Yes, the product works really perfect in E. coli system. Actually, the first generation of the Strep-Tactin STREPTAG purification products were invented for the use in E. coli. And in E. coli systems, as you know, you have usually much higher contamination concentrations because you have the lysate. But For the most purifications we did here over the last decades, we get more than 90% pure protein in one-step purification process, even for E. coli systems. So you can use it for E. coli, for yeast cells, you can use it for insect cells, you can use it for mammalian expression, you can use it for membrane proteins or secreted proteins or proteins from lysates. Actually, you can do all these things with the system.
And if there are no more questions, I would like to once again thank Dennis for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2019. Please share that with your colleagues who may have missed today's live event. Also, stay tuned for our next presentation, a comprehensive workflow solution from gene synthesis to purified proteins, presented by Dr. Michael Liss. You won't want to miss it. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.